Αγαπητοί συνάδελφοι, το επόμενο τραπέζι είναι αφιερωμένο στη οφθαλμική γενετική. Θα μιλήσουμε για τις καινούριες θεραπείες. Έχουμε προσκεκλημένο από το εξωτερικό τον καθηγητή Λερουά και για να διευκολύνουμε λίγο την κατάσταση, όλη η συνεδρία θα είναι στα αγγλικά. Έτσι. Οπότε θα ήθελα να αρχίσουμε με τον πρώτο ομιλητή. We'll start with the first speaker, Mina Matavtsi. She's very well known to everybody. She's been a member of the board for many years. She's a professor of ophthalmology at Aristotle University. And she will be talking to us about genetic eye disease in a child, the long and winding road to diagnosis.
Good morning, everyone, and good morning, Professor Bartleroy. We're very happy to have you here, and it's an honor to discuss with you. We decided to conduct this session in English so that you could hopefully prompt in with your input. So, to everyone, if something is unclear, please stop me and ask. I will try to be as clear as possible. Genetic eye disease in a child is a difficult thing. It's a long and winding road, as Beetle said, for both the doctors and the family. I will start with a little girl that I first saw when she was 2 years of age. She was wearing 6 diopters of myopic lenses in both eyes, prescribed by someone else. And I followed her up closely because her myopia increased rapidly. We just heard a session about myopia, but this is something else. Like at 4 years old, she had already 12 diopters and 1450. By 9, she had 17 diopters of myopia. So early on, I told the mother, this is not just myopia that your child has. This is something more, something else, and we need to find it out. It turns out that Calliope's mom is a single parent. She has another daughter, 2 children to bring up. She has to pay for the glasses. Every 3 months we have to bargain which lens we're going to change because it's a very high cost. So when I told her about electrophysiology, which is not available in the hospital, so she has to go and pay for it, and genetic testing, which is not covered by the insurance company, so she has to go pay for it. That was a difficult thing to achieve. Luckily, the little girl developed fairly good vision. This is in logmar. In decimal, it would be 5/10 in the right eye, 4/10 in the left eye. And eventually, Calliope's mom gathered the money and had a genetic test in a local private laboratory, which turned out, brought back the result of a pathogenic variant, what we would call traditionally a mutation, μετάλλαξη, in the RPGR gene, which is a gene responsible for retinitis pigmentosa, something Calliope luckily has not developed so far. This variant is also present in another family which also had high myopia. I'll spare you the details. This is all to say that symptoms or signs of genetic eye disease in a small child or an adolescent cannot be so obvious from the start off. High refractive error is something we should all look out for because we face it in our everyday clinics. And we just have to think about, could it be something else? If you have the suspicion, that's the very first step that is essential to go to diagnosis. Then, of course, if you have nystagmus, you would think of a genetic disease. If you have an unexplained low visual acuity, if you do everything you can and the acuity does not increase in a child, if and this you have to bring out with your own history. You have to ask the questions. If the child cannot see as well at night or in mid-light, like something like the room we have in here, although it's fairly bright. If the child has difficulty at nighttime, cannot see the stars in the sky, you have to ask. And then you have to suspect a genetic disorder. If, on the other hand, the light is too much to handle, if there is an aversion to light, then you also have to think of a retinopathy. And then, of course, in a child, we don't just look in the eyes, we look at the systemic features, also in any patient, really. So we have to suspect a genetic disease if there are systemic features as well as the ocular signs. So the aim is to have a molecular diagnosis, because very often a genetic disease will present with a normal fundoscopy. It will not be far obvious to everyone, retinitis pigmentosa fundus. And then it's a marathon. It's not a short distance run. You have to be patient, you have to put the time in, but you also have to go down the right path to get to the end. Because if you get a diagnosis, then you will be able to follow up the patient the proper way, like look at the other systems apart from the eye. And you can tell from what we know already about the specific gene, the specific disease, what the prognosis is, and that's very important in a growing child. And then you can also support the family by linking them to other families that have the same problem or a similar problem. And of course, and that's the end of today's session, you can also perhaps talk about treatment. So far we only have one, but Professor Leroy will tell us all about the new treatments that are coming up. And this really forces us to give the patient a diagnosis, a molecular one. So it's very difficult to look at a fundus or to look at a patient and say, this is RPGR gene. It takes an enormous amount of time among the very best of experts who have dedicated their professional lives into looking into patients with different patterns. And then perhaps with this experience, you can start judging from the clinical appearance what the gene might be. And that is very difficult for us because we are general ophthalmologists or pediatric ophthalmologists, and even if we get to see some patients, we don't have the critical mass to detect a disease just by looking at it. We have to run more tests. We have to phenotype our patients as well as we can. And how do we do this? First, we take a very good history. We take a very good family history. That will direct us. We have to examine the patient. And if we cannot, we get the pediatrician to do it for us. We have to examine the eyes, and we have to examine the relatives. And all of these little clues will perhaps direct us to a possible list of possible diagnoses. And then just a little note, when it comes to low vision patients, it is very useful to test the visual acuity with the ETDRS, with a logmar scale, because then you can quantify the visual acuity, even if it is below 1/10 decimal. So there is a list of tests that we can do. It is not always possible to do them in a private office because we don't all have access to autofluorescence or OCT or fluorangiography, but we have to think about those tests because they might be necessary to give us clues. And electrodiagnostics I will not go into because it's a whole separate introduction by my colleague later on. But then all of this deep phenotyping will give us clues about genotyping, which is the ultimate test to use to reach a diagnosis. And there's a list of genetic tests that we have not been educated about in our general ophthalmology training, but we need to get little by little familiar with, so that we can communicate with the geneticists who run these tests and can help us, collaborate with us to manage the patient about. So this is a recent paper run on a number of European countries to see what genetic tests are done, how they are paid, who they are paid from, what they cost. And unluckily, Greece is not on that paper, and this is our fault. We need to find a solution to provide our patients with access to genetic testing. And I hope that our discussion may give us some clues later on with regards to this, because it is essential. Now that there are treatments for inherited retinal disorders, for genetic eye diseases, we have to educate ourselves and be able to help our patients with regards to this testing. So a molecular diagnosis is not easy. We don't often know which test to do, what the result means, and how this translates for the patient and their lives. And there are a lot of ethical issues about it. So it is very important, as today's message, that this is not a blood test that you order. You have to discuss with the patient before you do it and after it is done. The psychological cost is not zero. Some patients have high expectations. These patients are very dependent on getting a result, and sometimes they're disappointed, or they may feel guilty that they are the healthy ones and their brother or sister or mother has the disease. So it is a very complex issue. It takes time, it is costly, and sometimes the result is of unknown significance so far. It has to be updated, re-evaluated a few years down the line when we know what this variant of unknown significance, perhaps we know what it means, if it is pathogenic or not. We need time. And of course, it is very easy to say that, but nevertheless, we need to do the testing, because it's a very heavy burden for these patients to not know what they have. And sometimes the outcome is what we call actionable, which means that we are able to do an action about it. And we need to proceed with that, even if there is a risk we will not get an actionable outcome. Thank you very much for your attention.
Thank you very much, Mina. We'll go through the talks and the presentations, and we'll have some minutes at the end for questions. So our next speaker is Stavronia Koukoula, well known to all of us. Stavronia is an ophthalmologist specialized in visual electrophysiology. She's based in Thessaloniki at Ophthalmica Institute and in Athens at OMA Institute. We are ready to hear you. She's going to show us some clinical cases.
Clinical cases, yes. Good morning, everybody. Thank you so much for the kind invitation to take part in this Congress. Thank you, Professor, for being here and share your knowledge with us. And I'm going to show you some clinical cases, emphasizing on what Mina said and giving my personal perspective. Just to remind you that inherited retinal dystrophies or disorders is now the leading cause, the main cause of vision impairment in children and working-age adults. We know that it's a group of genetically and clinically heterogeneous disorders, and you see all these circles with names inside, and the well-known RP, retinitis pigmentosa, but all the others around. We know that diagnosing patients is really a hard time. It's challenging. And molecular genetics nowadays, the last 10, 20 years, gave us a better understanding of the diseases, a better understanding of the patients, and helped us with new therapies, with new therapeutic alternatives. And we have moved from talking with phenotyping and diseases to do a discussion about mutations. So again, I will come back to the challenges that these cases have. And the first challenge is the precision diagnosis. As Mina said, we need the precision diagnosis. We need the molecular diagnosis. But it's not only this. It's, we have to answer questions about if this is something stationary or progressive, of what is the prognosis, what should we do in a follow-up, what is the management of each one patient, a proper genetic counseling, and a counseling for their everyday life. So in this perspective, I'm going to show you a couple of cases in our genetic clinic. It's clinical cases, but I'm going to present it as clinical stories. So case one, a mild phenotype over a serious diagnosis. So this boy, 8 years old, came to us with a mildly reduced central vision. It was in a school check. No other issues. He has 8/10 in his right eye and 9/10 in his left eye. Otherwise, he was normal, and there was a referral for electrophysiology. So we have done VEPs and full-field ERGs, and I'm not going to go any deeper to electrophysiology. Just want to say that apart from a weird morphology in the VEP, implicit time and amplitudes were normal, and the full-field ERG were normal. So we moved to imaging, and maybe you already see this vessel across the macula, and the weird appearance of the fundus autofluorescence with the lack of the darkness on the macula again. And I think the answer was given by the OCT, where I think it's obvious to everybody, this foveal hypoplasia. There is a big list of the reasons for foveal hypoplasia, but we're not going to spend our time. We gave a gene test, and the answer was a mutation in the tyrosine gene responsible for oculocutaneous albinism. So it's a type of albinism. So what we have to say to parents is that it's something stable, and they don't have to worry about something else if something is getting worse or progressive. So we gave the answer, we gave the prognosis. Second and third case, there are 2 children with night blindness, and let me show you. The first, it was a girl 6 years old who came with nyctalopia and reduced, she had problems with peripheral vision, mommy noticed, of course. No other family history, but she wears a cochlear implant because of neurosensorial hearing loss. So good central vision. And let's move to the imaging. I think you all saw the weird retina, like salt and pepper appearance. And in autofluorescence and OCT, this is more obvious with this hyperfluorescent ring on the fundus and the disturbance of the photoreceptor layer in the periphery of the posterior pole. And we have done electrophysiology, which is unfortunately undetectable in both scotopic and photopic ERG, which is in keeping with road cone dystrophy. And these are the visual fields, and I think that everybody has a diagnosis in the mind, which is retinitis pigmentosa. But it's not only that, of course, because we know that this girl had neurosensorial hearing loss, so the diagnosis is Usher syndrome type 1B, which means that it's, let me remind you of the Usher syndrome, that it has 3 types, and our girl is type 1, which is the more severe prognosis of the 3 types of Usher syndrome. So when I saw this girl, I know by default that it was something severe, and I had a discussion with mommy that she will have issues with the peripheral vision, which was, confirmed with the follow-ups, and we already have discussed from this first time that it's a severe prognosis, that she will need help, and maybe she will need to make an everyday life education with white cane or whatever else help her. Having said this, another guy, a boy, 9 years old, he was again diagnosed with sensorineural hearing loss. He wears a cochlear implant. He rather has some issues with night vision. He has good vision in his left eye and 6/10 in the right, maybe amblyopia. And he has rather normal fundus. But he was diagnosed with Usher type 2. And he came to us in order to see the status of the disease. So the autofluorescence was close to normal, and the OCT has some disturbances, but again, it was close to normal. And the full field ERG we have done, it's in keeping with rod cone dystrophy, but again close to normal. He had some issues in the implicit times, but amplitudes were really close to normal. So we have a guy with Usher type 2, which has a better prognosis. And at that time, I didn't have to say that to the parents that they have to concern unless something happens. But he's going to worsen, but not now, let's say. He will go to adulthood, and then he will have some issues. So we saw 2 children with the same, let's say, name, with the same diagnosis, but entirely different status and entirely different prognosis and future. And I'm going to finish with 2 other girls with reading difficulties. And the first girl, 10 years old, with school problems, reading difficulties, central vision problems, 1/10 the visual acuity, and I think that you already saw the flecked retina, which is more obvious in the fundus autofluorescence and the macular atrophy. The full field ERG revealed the cone rod dystrophy, which means apart from the diagnosis of Stargardt disease, which is a molecular diagnosis, we have categorized the Stargardt in Stargardt type 3 because of the full field ERG, because of the cone rod dystrophy. So it's a severe diagnosis. We know that this child will have peripheral vision issues, and again, she was really one of the best pupils in the school. She learns music, but she really has issues to navigate with her peripheral vision, and we have to discuss this with the family, and we have to give our advice. On the other hand, another girl, 10 years old, again, same, let's say, history, 2/10 the visual acuity, and I'm showing the fundus autofluorescence with this hyper autofluorescent ring in the center and this gap on the fovea. And the full field ERG was normal, so normal scotopic and photopic function. The pattern ERG is abnormal, of course, as expected. And again, this is a Stargardt disease, but it's a Stargardt type 1, according to the full-field ERG. So it's a better prognosis. I've been followed her a couple of years, so this is her progression. I mean, around 10 years after the first prognosis revealed the typical appearance of Stargardt disease. And again, she's pretty much well with her peripheral vision, as electrophysiology informed us. She went to the university, she studies English literature, and she lives in another town on her own. So she had no issues with her periphery, with her navigation. To conclude, I just want to emphasize that inherited retinal dystrophies need from us a precision diagnosis. It's not only phenotype and genotype the patients, but it's the status of the patient, which allows the patient to be aware of the capabilities and the limitations they have, allows us to support and management the patient, and of course, drive them to the new treatments. Thank you so much.
Thank you very much, Stavronia, for the beautiful presentation. And our next speaker is Evi Panayotou, who is a new member in our society. Evi is an ophthalmologist based in Thessaloniki. She's done her training in ophthalmic genetics at Ghent University with Professor Bart Leroy. And currently she's based at the HEPA hospital, but hopefully she will be coming to Athens as well, at least partly. She's now going to talk to us about Luxturna gene therapy in pediatric patients.
Thank you, Gathi, for this invitation. It's a great honor, and it's also a great joy for me to have my mentor here, Professor Leroy. So I'm going to talk to you about Luxturna in pediatric patients. The therapeutic landscape of IRDs is wide, but the type of therapy that has been most advanced is gene supplementation, and Luxturna, which is the only approved therapy so far for IRDs, belongs to this category. And it focuses on patients that carry mutations in the gene RPE65. The gene RPE65 produces the protein RPE65, which is expressed in the retinal pigment epithelium and has a key role in the visual cycle. Patients that carry biallelic mutations in this gene have commonly a severe retinal dystrophy that may vary from Leber congenital amaurosis to retinitis pigmentosa. They have congenital night blindness and often nystagmus. Their fundus looks fairly normal in the initial stages, but later it resembles retinitis pigmentosa. By the age of 18, more than half of these patients have a visual acuity of worse than 1/10. What's important here is that we see this structural functional dissociation, which means we have this relatively preserved retina, as you can see in this example, but with a disproportionately severe vision loss. And this is crucial because it gives us a window of opportunity to treat these cells that are dysfunctional, but they're still there. So Luxturna is actually the commercial name of voretigene parvovec, and as I said, it's a gene supplementation therapy that replaces the defective gene by delivering the normal copy of the gene into the cells, and this requires viable retinal cells. The preferred method to transfer the gene into the cells are these adeno-associated viruses. So it took a long time from the initial trials in animal models until its final approval. It took 20 years. But let's see the pivotal trial that led to the approval. And what is this voretigene parvovec? It's a surgery that includes vitrectomy and subretinal injection of 150 billion copies of this viral vector that carries the normal copy of the RPE65 gene in a volume of 0.3 ml. And in this way, we create a localized retinal detachment, which we call bleb, and this is absorbed within a few hours. So after that, what we expect is that the virus, the viral vector, will enter the RPE cells. They will deliver the normal copy of the gene into the nucleus, but the normal gene does not integrate in the genome of the patient, but stays separately in an episome and expresses the normal RPE65 protein. So in this trial, what we needed was a meaningful outcome measure that would reflect daily living of these patients that have low vision and night blindness. And this is why this multi-luminance mobility test was developed. And in this test, we test the speed and accuracy of a patient that tries to navigate in a maze, which is basically a room with different obstacles under different lighting conditions. And the second outcome was FST, full field stimulus testing, which is also for patients with low vision who cannot fixate, and it can measure the total retinal sensitivity to light. So in this trial, what we saw was a significant improvement in both these measures, in the mobility testing and FST. And this improvement was seen in the intervention group and started from month 1 and persisted until the end of year 1. We also saw an improvement in visual field, a non-significant improvement in visual acuity, and generally a good safety profile that was consistent with the surgical procedure. So after the approval in 2017 and 2018, we continued following up these patients, and we saw that the phase 3 patients showed a persistent benefit up until year 5 in the intervention group and year 4 in the control group, because the control group got the treatment 1 year later. We are soon expecting to hear about the outcomes at years 8 and 9, but we also know that the patients that participated in the previous trial, phase 1, in these patients, the FST improvement persisted up to 7.5 years. But what about real-world outcomes? The PERCEIVE study is a post-authorization study that is still ongoing, but its most recent report included almost 300 patients from 24 countries. Almost half of them were pediatric, and we saw indeed that their FST is significantly improved. Sadly, the mobility testing is not available in all centers, so we don't have this data. And the visual acuity was, again, not significantly changed. But that's not a surprise, and it's not a failure, because in these patients, their vision is very low in most of them from birth, and amblyopia has to play a role here. And in addition, RPE65 is more important for the function of the rods instead of the cones. In the safety section, we did see some chorioretinal atrophy that I'm going to talk about in a minute. But there are a few more smaller studies of real-world outcomes. They do have heterogeneous outcomes, and the patients' numbers are small. Besides, this is a rare gene. But if we try to focus on the pediatric patients, we can see this German study with 5 kids over 6 years old. The visual acuity did not significantly change, but the low luminance visual acuity, which is a visual acuity with a filter, and it represents vision in mesopic conditions, it did significantly improve, along with the FST. Also, a couple of more studies actually showed that the visual acuity may improve in children by 1 or 2 lines even. And when this was compared to adults, we did see that children perform better after treatment. Now, if we look at even younger children from 3 to 7 years old, again, we can see a significantly improved visual acuity and FST. And for the younger ones, when their parents were asked, they did confirm that their children were more independent and more confident and could better navigate in dim light. So what was this chorioretinal atrophy? We have started seeing this in some patients, and this is atrophy that exceeds the natural course of the disease, and seeing atrophy at the injection site is something expected, but sometimes we do see it within or even beyond the treatment area. Now, we do not know why this happens. Could it be the surgical procedure or inflammation or toxicity? But what is really interesting is that the patients that benefit the most from the treatment, they are more prone to develop this atrophy. And these patients are mostly school-aged children and young adults, as you can see in the blue color here. And again, in these patients, they have a great improvement which is not affected so far. So in conclusion, Luxturna works. It's not better, but it's not perfect, but it works. It seems that treatment in younger age may be preferable. Our findings so far are consistent with clinical trials, but we do still have a lot to learn. And I would like to finish with these videos, which were kindly provided to me by Professor Leroy. So this is one of his patients, around 10 years old. She participated in the clinical trials, and on the left you can see before treatment this mobility testing where she has a real difficulty to navigate. She makes mistakes and she takes a lot of time. But on the right, this is 1 year after treatment. You can see how quick she is. She doesn't make any mistakes and she completes the task successfully at the same low light conditions. Thank you for your attention.
Thank you very much, Evie, for the beautiful presentation. And dear colleagues, the time we've all been waiting for has arrived. I'm honored to present to you Professor Bart Leroy from Ghent, Belgium. Professor Bart Leroy is Professor of Ophthalmology, Ocular Genetics, and Visual Electrophysiology at Ghent University Hospitals, and also he's the head of the Department of Ophthalmology at Ghent University Hospital. He's a world leader in genetic clinical studies, and he was part of the team which developed Luxturna. Currently, he works hard to bring together patients with IRD, dystrophies, clinicians, and regulatory advisors, agencies, so that more genetic therapies can be approved. Professor Bart Leroy, thank you very much for coming. We are happy to have you here. You can start your talk, your presentation on ocular gene therapy beyond Luxturna.
Thank you, Agati, and thank you also to, I think, 3 people whom you've seen are Greek. Yes, they're all from Thessaloniki, but they're superb in ophthalmic genetics. And Greece and Greek patients deserve the same approach as anywhere else within the EU. And so I think that Greece needs to up its ante in getting ophthalmic genetics up and running more and using the services, amongst others of 3 individuals whom you've seen speak this morning, and who have the inherent quality to make waves for Greek patients. You all can, actually. And so I'm very grateful to be here. I love Greece, I love the Greeks. And Athens is obviously always a very attractive place to come to. My brief is to talk to you about stuff. Is Luxturna the end, or where are we going? And so I'll be talking to you about a couple of things that go beyond Luxturna. I just want to show you this. This is not to impress you. It is very important to me to say to you that I do not take money for consultancy myself. I reinvest that in research by paying 4 clinical trial coordinators with the money that comes in through my consultancy services. The 3 companies that are encircled are companies whose products will feature in this talk. So we know that humans are made up by a set of 20,000 plus genes from dad, and again from mum, the same thing. And currently, if you count, there are 335 genes involved. If you Google RetNet, you'll get to the site that keeps track of how many genes are involved. It's done by Steve Dager from the University of Houston in Texas. Currently, as you can tell from this list, for Leber congenital amaurosis, which is the group of conditions where light doesn't get translated into electrical signals. That's what it really is. We have 24 genes and counting, because only 70% approximately of our patients are genetically accounted for. It forms a continuum with early onset retinal dystrophy, Leber congenital amaurosis, and so there are a couple of overlaps genetically. So if you think about administration routes of something that you can put within the bag of genetic therapies, you can actually go intravitreal, which is easier, as we all know as eye doctors. You can go subretinal, as Avi was showing, which an approach we've used.
Καλημέρα σας. Καλωσορίσατε στην Photovision Art. Σε 10 λεπτά από τώρα, στην αίθουσα Α, θα γίνει workshop με εισηγητή τον Μάριο Θεολόγη με τίτλο The Art of Human Body. Ευχαριστώ.
I don't know what
We
she said, but anyway.
for this. It was not in the program.
So I hope I can continue. Avi was showing a subretinal approach, which we've used for Luxturna, and it is doable, but it is quite a bit of a surgical approach, much more complex than is the intravitreal route. And suprachoroidal injection is currently being evaluated. It hasn't been used in the clinic for IRDs. So I'm going to give you 3 examples of where we're going. I do not, in this short time, want to be complete. Gene augmentation therapy for Gucci 2D, if you're a fashionista, otherwise you can say Juki 2D. Leber congenital amaurosis, which basically is due to that fuchsia protein that brings back a photoreceptor from light stimulation for it to go again. So if you don't have a properly working Gucci 2D gene, you don't have proper proteins to bring back your photoreceptor from photoexcitation. And so you always do that. We continuously do photoexcitation, going back, photoexcitation, going back, and this protein is essential. And so very, very recently, Paul Yang, who works at the KCI Institute in Portland, Oregon, has published this important paper on a gene therapy for Gucci 2D related Leber congenital amaurosis. And the essence, I'm not going to go into the details, but the essence is that they used another type of adeno-associated virus than Luxturna, which is AAV2. They're very similar in structure, and 15 patients were treated with basically fairly good results. So they divided it up in 3 doses. It was a dose escalation phase and a dose expansion phase, then included 3 adults and 3 pediatrics at high dose. And basically they looked at treatment emergent adverse events as outcomes. So this is a phase 1 study. You go for safety first, but obviously as a secondary, you also go and see whether anything works. This is the structure, but not very important. And so those patients who received a high dose at month 12 had a mean change of the FST value of 20.3 dB for treated compared to 1.1 dB for untreated eyes. Now that may not tell you anything, but it's basically flashing a light into a dark adapted patient and then seeing when does the patient react. And the patient has to subjectively react. But 20 dB is huge. It is an enormous change in sensitivity, and it's certainly considered meaningful. So this is the first, a phase 1 for Gucci 2D, which is a second trial after the RP65 work that's been done with Luxturna. Let's change gears and go for something completely different, also a genetic therapy, but we're now talking about an antisense oligonucleotide. And that may sound like Chinese to you if you're not into genetics, but basically it's a little Velcro of RNA. So you build an RNA molecule with 17 nucleotides, and you inject that nakedly. There's no carrier, no virus, no liposome or anything. You inject that nakedly, and it has an effect. Now let's first look. CEP290 is that orange protein. The protein works in the connecting cilium. As you probably know, proteins that are involved in translating light into electrical signals in the photoreceptor outer segments need to get there because they're being produced just like any other protein around the nucleus of the cell. The connection between cell body of the photoreceptor and the outer segment where electricity is made out of light, that connection is the connecting cilium. CEP290 plays an essential role in shoving the proteins down that connecting cilium so that it can happen. What? Vision, translating light into electrical signals. Now these people, artificially colored in blue on the left-hand side, have photoreceptors present on OCT, even if they sometimes have no light perception. So there's potential there, but it simply doesn't work. Generally speaking, it's a very severe phenotype, the CEP290 related Leber congenital amaurosis. There's a high unmet need for this, and what we've done in a phase 1, 2, and then a 3 is inject that little Velcro of RNA intravitreally. Now, how does that work? In this gene, a large majority of the patients have one specific mutation, at least in one of their 2 copies. You need 2 bad copies to have the disease, otherwise you're an unaffected carrier. So parents don't know they carry it, and they throw the bad gene in, together into one child, and the child has one of the 2 mutations that is very frequent in Europe. Sometimes they're homozygous, so they have the frequent mutation in both copies. That mutation induces a screw-up of making proper RNA. So you have DNA, exon, intron, exon, intron, exon, intron, which is coding text, white block, coding text, white block, coding text, white block. That's the structure of the DNA. That gets transcribed into RNA, first premature messenger RNA, with the same structure, coding, non-coding, coding, non-coding, coding, non-coding. Then splicing happens, cutting and gluing together, so cutting and pasting so that only the translatable text is in one message, and that's the mature RNA. The mature RNA can then be translated into something completely different, which is the protein. With that mistake in intron 26, which is the non-coding part 26, the machinery gets screwed up, and the message of intron 26 stays into the mature messenger RNA. The protein is made, it's far too short a protein, and the patient is blind because the protein is worth nothing. So what we did, we injected a little Velcro of RNA that glues itself against exon or intron 26 so that it gets corrected. So I'm not going to dwell on the details, but what we did was we did a phase 1-2 trial, and it seemed to work. It was safe. We had some cystoid macular edema in some patients. We had cataract in some others. We said, this works well. We said, let's do a 2-3 trial. So you do a phase 1-2 first, and then a 2-3, and with a phase 3 trial, you can go to the regulators and say, do you think that this is proof enough for us to introduce it to market? And that's what we did. The discussions with FDA and with EMA were such that FDA did not allow us to do an intra-person comparison. So we couldn't treat one eye and then take the other eye of the same patient as a comparison. And that was a mistake. FDA told us, you have to take one eye of one patient, treat that eye, have another patient, unrelated, sham treat, that is, act as if you treat one of his eyes or her eyes, not treat the second eye, and then compare those two eyes, the treated eye in one patient, the sham treated eye in another patient. That doesn't work. It doesn't work, and I don't have the time to go into details, but suffice it to say that two patients are never the same, especially if they're badly sighted. Plus, one day is not the other day. We have redundancy of our visual function. I have a little cold now, my vision's perfectly fine. If somebody who's badly sighted has a cold and doesn't feel 100%, they'll have lower vision. If they haven't slept properly, they will have lower vision. And so our comparisons were not what was suggested by the questionnaires that we asked the patients. We asked the patients, what do you think of the treatment? And they all said there is improvement. And I'm not going to go into the details, let me go through this, but what we had was the following. We said, no, we should do another analysis. We should actually make sure that the interpersonal differences are minor. And so what we did was, suggested by the European Medicines Agency, FDA, Food and Drug Administration, had asked us the other approach. EMA said, why don't you take the best corrected visual acuity, for example, of the treated eye in patient 1, you take the best corrected visual acuity of the untreated eye in that same patient, you deduct the second from the first, you normalize the result, you do the same for the other patient who is sham treated, and then you compare those deltas. And then we had significance. And we said to the regulators, can we now introduce it to market? And they said, well, no, that was not the goal from the outset. You'll have to redo a phase 3. The company that was involved was a small Dutch startup. They pulled out and they threw it in the bin. Until a French company, Thea Pharma, I think Thea Pharma is on the Greek market as well, is that correct? So Thea Pharma is a French company in Europe, and Thea Pharma started a new company called Sepul Bio. They picked this antisense oligonucleotide treatment up, and they're now planning a new phase 3 so that we can get this to market. To end with a little dessert, AAV2 was what Avi was talking about. RP65 is a small gene. I have been speaking the first part on Gucci 2D, small gene fits in AAV. But AAV is like a small van, a small delivery van. For some of the genes, the Usher genes that people spoke about before me are large. The Stargardt gene is huge. So you can't get it into AAV. So what do? People have used lentivirus. That doesn't seem to work. So AAV can be leveraged by doing the following. You can use 2 viruses. You basically do the following thing. You go buy a sofa for 2 people in IKEA, and you do that with a Fiat 500, and you try and transport it. That's not going to work. You have a friend who also has a Fiat 500. You cut your sofa in 2 halves. You put one half in one Fiat, the other one in the other Fiat, and you drive the one after the other. You deliver together and you put it back together. That's the philosophy of dual vector technology. So there are 2 types of, two companies that are really investing in this. First, for Usher syndrome type 1B, you've seen one of my colleagues present a myosin 7A case, and that is now ongoing. The first patient has been dosed in Napoli in Italy, and this is Avantgarde Bio, a spin-off of the University of Napoli. And for Stargardt disease, they're planning a trial too. There are 2 different approaches. You can do concatemerization, which basically is 2 DNA molecules that recombine, and then that recombined DNA molecule is being used to produce a full-length protein. You can also make 2 intine-based technologies, and it's far too long to go into the details, but what you get is 2 proteins, and those 2 proteins will recombine. The one or the other, there are some differences in how to do this, but it doesn't matter too much. And this is Splice Bio. The first two slides I showed here was Avant-Garde from Napoli. Splice Bio is a Spanish company that does also work with the protein approach. So they make two halves for the Stargardt protein, and they will try and get to clinic in 2027. So I think I've flown with you through a complex set of things, but I think genetic therapies are hot. They are not perfect. My first patient who was treated with gene therapy was the first child ever to be treated in Philadelphia. I flew with him to Philadelphia. And the boy, after a week, said to his mum when he flew back, "Mum, I can see the city lights. I can't see them with my unoperated eye, but with my operated eye, I can see the city lights." Isn't that a change? He's now a psychologist, working as a psychologist. He's got a lot less vision than he gained, but his vision is still much better than before he was treated. I asked him, knowing that you would wane a little bit in effect, would you have it again? And he said, "Oh yes, by all means, because this allowed me to do my studies and to be a professional, and it really helps me in my life." Thank you.
Dear Bart, thank you so much for sharing all this information with us and making it easy for us to understand. Thank you so much for that. So now we have time for some questions, if there are any. If someone would like to ask something. Evie, would you like to ask something?
Yes. Can you hear me? Yeah?
Yeah, if you speak up, we can hear you.
So, Bart, you have huge experience from Luxturna. What do you think pediatric ophthalmologists and general ophthalmologists should tell patients and parents of pediatric patients about what to expect from Luxturna and in general these treatments? What impact would they have on their lives?
It's a very important question, Evie. I think if I can convince the crème de la crème of Greek pediatric ophthalmology to say the following, I'll be a very happy person. So don't expect these gene therapies and genetic therapies, however we approach this, with getting a novel copy of the gene in, like Luxturna and the Gucci 2D, or influencing the translation of what is DNA into what is the protein, to interfering with RNA. It doesn't really matter how. Do we make normally sighted people out of them? By God, no. We will not. We will never. And if you think that we will ever do that, then I think you need to think again. Why? Because we basically start off with retinas that are not normal. And so we also start off with people, and I don't have to teach this crowd how visual development works. You know this maybe better than me. But if you have bad vision from birth for a year, 2 years, 3 years prior to getting the treatment, there will be a fundamental impact on the potential ever to be reached with any therapy that you do. And so bring the expectations down to a level that is attainable. So under-promise and over-deliver is the best approach you can get. Will we improve visual function? Yes. The first patient whom we treated is actually a lovely lady from Australia who lives in Belgium with her British husband, and she was 40 the day I called her up and I said, "Do you want to be the first patient to be treated with the now commercially available and completely reimbursed, by the way, in Belgium, reimbursed Luxturna?" And she said, "Why would you take me? I'm an end-stage person." And I said, "Because you've got nothing to lose, and you are a smart cookie, and I don't have the feeling that I'm pressurizing you into getting the treatment. And I still think that you will gain from this treatment." What did she gain? She said afterwards, "Bart, I can now see When I'm preparing my son's sandwiches for lunch at school, I can see what I'm doing on my plate. Before, it was just by touch. Is that good enough? I think it is good enough. Did she gain a major improvement in best corrected visual acuity? Oh no, light sensitivity did go up, yes. And that's what she got, seeing the sandwiches and seeing what she's doing. Seeing a ball that her boy kicked to her so that she could kick it back to him. Does that mean something? I think so. So we shouldn't promise normality by all means, but do you sign on the dotted line for improvement of vision or stability of what you have now? So we've spoken mostly about pediatric cases. For example, if you talk to an RP patient who's 20 or 25 years old or 40 years old, they'll have a super narrow visual field and they'll beg you, can you please stop the incremental change that I see on a yearly basis? Because it basically kills me. If you give stability or even bring down the speed of degeneration, you will also be very successful. So under promise and over deliver. That was too long an answer, sorry.
Thank you for clarifying that. It seems that stability is also important, and really genetic therapies change people's life. It's not necessarily to see, as Professor mentioned, improvement, but even stabilization is equally important. I'm just wondering if there is any other question from the audience, or Mina, if you would like to ask.
So the longest follow-up. So it's a very good question, Mina. The longest follow-up that I know of, I don't know the Napolitan patients who were the first treated in the phase 1. I don't know of them. And I haven't spoken to my colleague from Napoli recently. They were still doing fairly well. Yannick, I can say his name, the psychologist, because he's all over the press. He signed an agreement that we can be open about him. Yannick Duet was 9 when he was treated. He's 25 now. And so that's a long period. We haven't published those data yet because we have an issue. Like Avi was saying, we are expecting the new results from the years 8, 9, 10, basically. We have trouble because COVID came and we don't have many patient visits. So we're in trouble. We actually will have to jump. But he's been treated at 9 and then treated at 11 in his second eye, and he's 25 now. I see atrophy in the treatment area. I see atrophy in the periphery. So we haven't been able to stop the disease. Why is that? 300 microliters just treats your macula. I always say, well, what do you think? If you have a degenerate retina and the larger surface area, the periphery, is untreated, do you really think that you will have a major impact and a stabilization of the disease by just treating the center? And I don't think that we do. So the progression of disease beyond the treatment area, I think, also influences the result that you get in the treated area. But he's still much better than he was as a kiddie before the treatment.
Okay, thank you very much. I'm just wondering whether in the current genetic studies that you are running, if there are patients from other countries and would be interested in taking part, would you accept, and would be something like that feasible? Would be
we do.
Possible?
Yeah, we do. So we've set up a trial center about now 7, 8 years ago. And so we work with 4 clinical trial coordinators. We are now setting up, together with Sepul Bio, the TIA Pharma spin-off, the new treatment trial with the antisense oligonucleotide. And we are, for example, will be including quite a few Slovenian patients, some Romanian patients. So we get referrals from countries that sometimes don't have the access to the treatments. What we've also seen is that sometimes it's better to concentrate the treatments in one center where there's experience, because obviously the deltas that you can attain are small, and if the deltas are smaller still because of variability between the centers where the treatment has happened, you're losing potential. But we do actually accept patients. So it's a shame that Greek patients don't have access to reimbursed genetic screening. We screen genetically. Nobody is getting this for free. You know what the cost is to see me as a geneticist? So I'm also a geneticist. I do genetic counselings myself. I do everything in one go. And when I see them, I can charge a genetic counseling, which basically costs the patient 8.5 euros. The rest is paid for by the National Health Service. The molecular screening can cost anything between 425 and 1,650 euros. That is, the latter one is the full whole exome sequencing with a lot of work, but the patient will only pay 12.80 euros, whatever test is being done. And so that is a fundamental right. You've heard from my colleagues why that is required. You can counsel the patients, you can take away the not knowing by, yes, you are at risk, maybe you can protect yourselves against the sun. The Mediterranean diet is the Greek diet, so that's the best diet you can have with all the nutrients you need. But genetic testing ought to be available for Greek patients. It's an absolute scandal that if we want to be an EU, that some of the countries have no access to genetic screening for a fair price. And I'm saying this to you because I think that you can change this all. I honestly think so. You can go and talk to the government. And if this powerful group of pediatric ophthalmologists of Greece, you represent quite a powerful group. If you take that into action, you can make a change. Thank you.
I'm sure that this is something that our society will try to achieve. Maybe we should all together exert some force to the government and see if we can change things for the best interest of our patients. Thank you very much for coming. Thank you very much.